Good morning to everybody. Today I'm going to teach you reproduction in flowering plants part 2, and under this I'm going to teach you double fertilization and post fertilization events and structure. So in case of double fertilization, fertilization, it is simply the fusion of male gamete with the female gamete. And in case of angiosperms, generally this is the major event that takes place. And here the fertilization will be two different types. Under this double fertilization, it includes two events. One is syngamy, another one is triple fusion. So in case of syngamy, the first male gamete that is in the pollen tube that fertilizes the egg cell, and the other male gamete that fuses with the central cell, and this second fusion, the male gamete with the central cell, this is generally called as triple fusion, as there is fusion of three nuclei, one nucleus from the male gamete and two nuclei that is in the central cell. So that is why it is called triple fusion. The product of syngamy, that is the zygote, and the product of triple fusion, that is pen. And both this syngamy and triple fusion, generally we call this as the double fertilization. Primary endosperm nucleus pen, it forms primary endosperm cell, and primary endosperm cell further transform into endosperm. And endosperm, it is the special structure that has reserve food in the form of protein, starch, your fats, and this nutrition by this endosperm is provided to the developing embryo, and that embryo is formed from the zygote. So you can see in case of this embryo sac, you can find three cells towards the telogal end, you can find three cells towards the micropylar end, and in the micropylar end you can find the two special cells having the filiform apparatus that can escort pollen tube, and they are the synergids, and in the synergids only there will be release of the two male gametes. And you can see the left hand side, the pollen grain germinates and there will be formation of the pollen tube, and this pollen tube, it has two male gametes and one tube nucleus, that is the nucleus from the vegetative cell. Then come to post fertilization structure and events. We are discussing first about the endosperm, and endosperm, that is the product of triple fusion. And endosperm development, it precedes embryo development. So can you tell why there will be development of the endosperm first, then only after that there will be development of the embryo? Because first the food will be stored in the endosperm, and when there will be further your development of the embryo, food will be made available to the embryo from the endosperm. So endosperm tissue, this is filled with reserve food materials, and that will be useful for developing embryo. There are three major types of your endosperm development, that is free nuclear endosperm development, cellular endosperm development, and halobial endosperm development. So in case of generally free nuclear type of endosperm development, that pen that divides and it forms large number of nuclei. There will be formation of more and more nuclei, but these nuclei are not laid down by any cell wall. There will not be any cell wall. So that is generally your free nuclear endosperm development. But in case of your cellular endosperm development, as we can see it in case of utricularia, in utricularia, pen it develops first, it makes two nuclei, and each nuclei further they gather the cytoplasm, and there will be two cells with the cell wall there will be two cells, and these two cells further they undergo both the karyokinesis, cytokinesis, again 4 cells will be formed. So this way whatever your nuclear division will be there, that is generally followed by the cell wall formation. So this is generally your cellular endosperm development. And in case of your last type, that is halobial type, in case of your halobial type of your development, in case of halobial type of endosperm development, you can see the last picture here. The primary endosperm nucleus divides into two, and further this nuclear, this lower one, you can see some nuclei, further division will be there, and some nuclei they will be forming the cells. Cell wall will be there. So you can see in the last structure, the lowermost one, there are some cells. There are some cells you can find. So these cells will be there, and you can find some free nuclei are there inside this embryo, inside this endosperm. So this is halobial type. And in case of your coconut water that you take, that coconut water is the example of free nuclear endosperm. And the white kernel, the edible white part that you take, that is a cellular endosperm. So this endosperm may be completely consumed by the developing embryo, like in case of your pea, groundnut, beans, before seed maturation, or it may also persist in case of some mature seed like castor, coconut, etc. And this is generally this endosperm, it can be utilized during germination. Then in case of your embryo, so first part it was the endosperm, that is endosperm formation. It is one of the important part under the post-fertilization event. Then come to the embryo. So you all know what is developed into embryo. Of course it is the zygote that is developed into embryo, and this early stages of embryo development generally we call as embryogeny. So this embryogeny, it is similar to both in monocot and in dicot. And the zygote generally it undergoes division, and it can form different forms of your embryo. First it can form the pro-embryo, then subsequently it forms globular embryo, heart-shaped embryo, and then the mature embryo. So in the early stage, when zygote, you can see the picture. So in the picture you can see first the zygote, that is single cell, then that develops into two cells. So in case of these two cells, one cell is terminal cell, another one is suspensor cell. So suspensor cell, it further divides and it can form a bigger suspensor, and the lower terminal cell that divides and it forms a lower part, that is the globular embryo. And this globular embryo further transform into heart-shaped embryo, then the mature embryo. And you can see here, in case of the long suspensor, this suspensor, do you know what role it can play? Suspensor here generally it can help in pushing your embryo into the endosperm. Okay, it can push that, the suspensor can push this embryo towards the endosperm, so that your embryo can get nourishment from the endosperm. We can find this embryo both in dicot, also in case of monocot. So in case of dicot, generally the dicotyledonous embryo, it has one embryonal axis and it has two cotyledons, and the portion of this embryonal axis above the level of cotyledon, you can see that portion we call hypocotyle, and below the level of your cotyledon, that is called as below the level, that is called hypocotyle, and above the level of cotyledon, that is generally called epicotyle. Okay, and the lowermost part you can see in case of this radicle it is having the root cap. In case of your monocot embryo, here you can find only one cotyledon, and this cotyledon is generally called as scutellum. It is generally situated at lateral side of the embryonal axis, and at the lower end of the embryonal axis it has radicle and root cap, and that is enclosed with a sheath. The lower part you can see it is enclosed with a sheath that is called as the coleorhiza. Okay, like that the upper portion where you can find the shoot apex, it has also few lip primordia, and that is enclosed in a hollow coleus structure that is called as the coleoptile. Then come to seed. So after fertilization, so in post fertilization, generally the fertilized ovule that is transformed into seeds, and every seed you can find seed coat, you can find cotyledon, and you can find embryonal axis. So in case of your seed coat, it is testa and tegumen that you already have gone through in class 11th. Mature seeds, it may be non-albuminous or it may be your albuminous. So non-albuminous or X-albuminous, in the mature seed you cannot find any albumin or any endosperm. So that is generally your non-albuminous, like your groundnut, pea, gram. So there you cannot find any your endosperm. So no residual endosperm is left in case of the mature seed like the pea or groundnut. So such seeds are generally called as non-endospermous seed or non-albuminous seed or X-albuminous seeds. But in case of your albuminous seeds, you can find some residual part of the endosperm that is not completely used up by your developing embryo, and generally we can find some amount of endosperm in case of wheat, in case of maize, in case of barley, in case of castor. Occasionally in some seeds such as your black pepper and beet, nucellus it remains, some part of the nucellus it remains persistent, and this residual persistent nucellus, this is generally called as perisperm. Then you all know ovule is covered by integument, outer integument and inner integument. So this integument in the seeds, they are nearly hardened, they form the top protective seed coat, and the opening that you can find the seed, a small pore, that is your impression of the micropyle. So this micropyle, it helps in facilitation of entry of oxygen, and that generally helps during the germination. So as the seed matures, water content that will be reduced and seeds they become dry. So it has only about 10 to 15% of moisture by mass, because if there will be more and more moisture content, there is chance of spoilage by microorganism. And in case of such seeds and such dry seeds, metabolic activities are also slowed down, and the embryo may enter a state of inactivity or suspended animation, that is generally called as the dormancy. And when there will be arrival of the favorable conditions, then again this embryo it become active and there will be germinations. Fruit. So in post fertilization, it is the ovary that is transformed into fruit, and wall of the ovary that generally develops into fruit wall, that is called as the pericarp. In class 11 we discussed pericarp, underepicarp, mesocarp and the endocarp. And when there will be maturation of this ovary, simultaneously there will be also withering or degeneration of other floral parts like the calyx, corolla or the stamens, and they fall up. And the thalamus, sometimes it contributes to the fruit formation, and when thalamus it contributes to the fruit formation, such type of your fruit we generally call as false fruit, that we find in apple, in strawberry, in cashew. But Most of the fruits, they however develop from the ovary, and they are called as true fruits, like your mango. Mango, that is the true fruit. So there are few species where fruits are also developed from your, they are developed from the ovary without fertilizations, and such type of fruits generally called as parthenocarpic fruit. As we can see in case of banana, certain variety of your grapes also you can find they are without any seeds. In case of your orange also some varieties you can find without any seeds. So such fruits without any seeds, they are generally the parthenocarpic fruits. And with the help of your hormone spray also we can obtain parthenocarpic fruits, and the hormone really it is the auxin, it can induce parthenocarpy. So you can see the figure here in case of this apple, in case of your strawberry. So you can simply see the your false fruits. So these fruits, they are generally developed directly from the thalamus. The seeds that is released after the dehiscence, seeds will be made available to outside. So how long do seeds remain alive after they are dispersed? This period generally it varies from plant to plant. In some cases this viability of the seed may be few months, in some cases it may be several years, or in some cases the seeds they may remain alive for hundred of years. There are several records of very old yet viable seeds. The oldest one, Lupinus arcticus. Lupinus arcticus, that was excavated from Arctic tundra. The seed germinated and flowered after an estimated record of 10,000 years of dormancy. And there is also another recent record of 2,000 years old viable seed of Phoenix dactylifera, date palm, that was discovered during the archaeological excavations at King Herod's palace near Dead Sea. Then come to your another your phenomena where there will be formation of seeds from ovule without fertilization. Such phenomena is generally called as the apomixis. So this apomixis generally we can find in case of Asteraceae, in case of grasses. So apomixis is a form of asexual reproduction because it mimics sexual reproduction. There are several ways of development of apomictic seeds. So we can develop apomictic seeds. Generally farmers they are using more and more hybrid seed. Hybrid seed can be used by farmer in one season. In the next season they cannot use because there is segregation of traits. The desirable trait which are made accumulated in the seed, that is segregated in the next season. So that is why they cannot use your hybrid seeds. But in case of apomictic seeds, if there is production of apomictic seeds, then there is no such segregation of characters in the hybrid progeny. So much research is there on this apomixis, so that more and more apomictic seeds can be made available for agriculture. And the last one, the polyembryony. So we all know that inside one ovule we can find one egg, and this egg is fertilized to form the zygote, and zygote further it forms the embryo. So basically we can find in one seed one embryo. But sometimes what happens, the nucellar cells that you can find below the integuments, that sometimes can develop into many embryo sac, and they develop into embryos. So such in such species like your citrus, in citrus fruits like lemon and orange, you can find occurrence of more than one embryo in a seed. So when you are squeezing a seed of lemon or orange, you can find many embryos. So you can see this diagram. From one seed you can find many saplings. So these many saplings, because they are inside one seed, you can find many embryos. So this is the case of polyembryony. Thank you all.